Hey folks, Dr. Gilchrist with lecture 24 coming at you. Before we talk a little bit more about what we know about long-term potentiation, I thought it was really important to kind of remind you a little bit about what long-term potentiation is before we move on. So long-term potentiation is the finding that with high-frequency stimulation, you will get an enhanced EPSP to a subsequent single pulse in the future. So if I give you a high-frequency train of stimuli in rapid-fire succession, multiple pulses, kind of like what we will find is that if I give you one more single pulse, the EPSP will be much larger and it will last for longer. And this is largely because of the different presynaptic and later and those postsynaptic changes that happen in both the early and the later stages of LTP. So as just a review of what typically happens, what you're looking at here is a standard synaptic transmission. So we've got our NMDA receptors. They're currently blocked by magnesium, so calcium cannot enter the cell. We've got our AMPA receptor that is a standard sodium-potassium channel that opens up when glutamate binds to it. So our presynaptic cell basically releases that glutamate. It binds to the NMDA receptor and it binds to the AMPA receptor. Now, once this glutamate binds, we will have sodium go inside of the cell and potassium will leave. And because we have this greater sodium influx than potassium efflux, we get a depolarization, which is all we pretty much need to kick out this little ion of magnesium so that calcium can start to enter the cell and exert postsynaptic changes. So here's what happens when that begins. So again, our magnesium's kicked out, it goes away. So calcium is now able to flow in through the NMDA receptor. It binds to calmodulin. It then binds to calmodulin kinase 2, which starts to form different types of protein synthesis. So this will create new AMPA receptors. It will create a variety of cellular changes. And over the long term, what will happen is that nitric oxide will be released, potentially, going back to the presynaptic cell. It's why we call it a retrograde messenger. It travels backward. And that nitric oxide will facilitate greater release of glutamate from the presynaptic cell. So in late-term, or second phase long-term potentiation, we're going to see not only postsynaptic changes, but those presynaptic changes as well. So here's where we kind of left off. Early phase LTP lasts a few hours. It's only postsynaptic effects, so we get enhanced AMPA receptors and the formation of new ones. Late-term, long-term potentiation can last several days, all the way to infinite. We'll see new AMPA and NMDA receptors, as well as dendritic spines, which increase the surface area and allow areas where glutamate can bind. We also get that retrograde signaling. Nitric oxide will increase glutamate release in the presynaptic cell. So this raises the question, if synapses can be strengthened, can they be weakened as well? We know that long-term potentiation actually strengthens a synapse. We have more receptors, we have more glutamate release, and we have greater thickening of the postsynaptic density. But it turns out that they can also be weakened. Not all synapses are meant to grow in strength, and it's actually necessary for some of these things to be weakened for memory to function properly. So what you're looking at here is a pretty interesting graph. So I want to kind of orient you to a few different things. So first of all, what you're looking at, I'd like to draw your attention to this horizontal line right here that is 0 on the y-axis. So this 0 is basically your baseline. This is what an EPSP would look like under standard operating procedure. This y-axis is the percent change in the slope of the EPSP in CA1, so part of that Schaefer pathway. So if it's above 0, that is what we would call a long-term potentiation. We actually have a heightened EPSP relative to baseline. If it's below 0, this means that we have a smaller EPSP relative to baseline, and this is what we would call a long-term depression. Now, on our x-axis, we actually have the frequency of stimulation of Schaefer collateral axons. So we're dealing with the hippocampus here. Now, what you'll kind of notice is that for higher frequency stimulation, anything over 10 hertz, we typically will get a long-term potentiation. So those are those high-frequency pulses. They need to be at minimum 10 cycles per second. However, if stimulation is below 10 hertz, there is no long-term potentiation. You instead get that long-term depression. So EPSPs will be reduced to subsequent single pulses. So let's compare and contrast the two. LTP is due to high-frequency stimulation. LTD is due to low-frequency stimulation. Both of these are dependent on NMDA receptors. Both of them are inhibited by AP5, which is an NMDA receptor antagonist. So it blocks the NMDA receptors. And it turns out that when you apply AP5, you don't get LTP and you don't get LTD either. We do see that LTP is associated with a large calcium influx. LTD is associated with a small calcium influx. And LTP is associated with both pre- and postsynaptic changes. LTD is only associated with postsynaptic change. Glutamate release doesn't really change in LTD because there are no presynaptic changes. What happens instead is that because of low calcium influx, the AMPA receptors downregulate and cut their numbers. Now, it has been suggested that this may be dependent on different types of NMDA receptors. We know that there are different kinds. Some of them permit a large calcium influx, others don't. But generally, LTD is not studied as much as LTP, and so not as much is known. Both of these are pretty common in the hippocampus. There are other regions as well, but they typically tend to not be studied as often because NMDA receptors are largely concentrated within the hippocampus. And there's some evidence that with LTP basically functioning as a cellular form of memory formation, so LTP can be associated with forming or strengthening memories. You can kind of think of LTD as a method of either weakening or possibly even forgetting memories. And there's some evidence to support this. So the next question, so LTP has been suggested as one of the cellular mechanisms of how we learn and form memories. And this is a pretty plausible hypothesis for how learning happens at the cellular level. It's pretty consistent with Hebb's rule. So we get these cellular changes and no new neurons are created. But what is the evidence that actually links LTP with actual learning and memory? So this is a case where using an animal model can help us understand the function of the hippocampus. So one of the things that we know is that the hippocampus, particularly in rodents, is really critical for spatial learning. So what we can do is we can try to block LTP in the hippocampus and see how that affects spatial processing in rodents. And there are two different ways that we can actually do this. We can do this through something called the Radial Arm Maze, and another method is through what we call a water maze or the Morris water maze. So what you're looking at here is a radial arm maze. Here is our rat. Isn't it cute? So this is a case where food is basically placed at the end of each arm. Now, what you'll kind of notice is that to be able to get all of the food, the rat has to go out on each arm, get the food, but then it has to go back to the center of the maze before it moves on to the next arm. Now, generally, the rat needs to be able to remember where they have been, so they're not constantly revisiting an arm they've already gotten food from. Now, here's what's interesting. The way that they do this, rats have pretty good senses of smell. So what they make sure they do in this radial arm maze, so that we know that it's memory that they're using rather than their sense of smell, we make sure that the food, it does not have an odor, so that all the rats can really do is use their memory to help them figure out where they need to get their food. So here's something kind of interesting. So I want you to kind of imagine, here's a kind of a layout of our radial arm maze. So here we've got food placed in these arms. These other arms are locked off. So the rat will travel to these arms and get the food, and then we basically remove the rat. At that point, we're going to put food in locations where the rat didn't get it. So we're going to see if the rat wants to explore those unvisited locations. And generally, what we tend to find is that in general, rodents have a preference for novel locations. They don't necessarily like to go back to a place that they have been. So what we do, the rat will go to the location. So we're looking at a rat that has a normally functioning hippocampus. The rat will go to all of the locations where it had not gotten food previously. So it's going to all of these new novel locations that it hasn't checked out before, and it's basically rewarded by food. Now compare that to a rat that has a hippocampal lesion. After this delay period, the rat not only goes to the areas that have the food, but it goes to the arms it's already visited, and in some cases, it looks like the rat is visiting more than once. So we can kind of see that for rats that have a hippocampal lesion, there is no spatial memory after this delay period. Now, something that's kind of related to this is the Morris water maze. This is another test of spatial processing. So the rats are going to swim in this little tub of murky water until they are able to locate a platform that's actually hidden beneath the surface. Now, you'll kind of notice that there are certain types of spatial cues that are located around the maze. What we tend to find is that normal rats will learn to associate the location of that hidden platform with cues that are placed at specific positions around that maze. So let's see what happens when you compare performance on this task between a rat with no hippocampal lesion and a rat that has had its hippocampus damaged. So here's kind of what this looks like. So we've got our pedestal that is hidden just below the surface of the water, and the rat has to swim around the maze to find it. So you can kind of see in trial one, it takes a while for the rat to swim around, and it initially finds it. Usually within 10 trials, a normal rat with no hippocampal damage, it's able to go right to that hidden platform once it's put in the water. Now, to compare that to a rat with a hippocampal lesion, as you can see, their swimming patterns look just as scattered and just as much in a disarray as it did from the first trial. So this kind of indicates, again, that the rat is not learning from its experiences and that the hippocampus seems to be really critical for aspects related to spatial memory. So here are a couple of other things that have been done to look at how LTP supports performance on these different types of memory tasks. So one of the things that we can do is we can inject AP5 into the hippocampus, which again is an NMDA receptor antagonist, which means that the receptors will not be able to work. And we do find that learning on the water maze performance is actually impaired when this AP5 is injected into the hippocampus. We've also had researchers create NMDA receptor knockout mice. In this case, these mice are genetically bred to lack NMDA receptors, and we do find that for that special breed of knockout mice, their learning is in fact impaired. And then finally, one other experiment, we have what researchers refer to as Doogie mice. These rats are genetically bred to have enhanced NMDA receptor activity. And so what we find is that there is more calcium influx into these NMDA receptors. And not only is learning enhanced, but hippocampal long-term potentiation is enhanced. Now, you might be curious why we call them Doogie mice. At the time that the Doogie mice were being studied, there was a very popular show on TV called Doogie Howser, MD. It was a TV show that was on TV for a couple of years. I remember watching it as a kid, about a teenager who was basically a genius and had already gone to medical school and was now a doctor. And you might actually know Doogie Howser as the once and future Barney Stinson. That is actually Neil Patrick Harris, who portrayed the character. So let's talk a little bit more about LTP and spatial memory. Now, this is a little bit more correlational in nature, but it does actually help support the idea that long-term potentiation supports memory and learning. We do tend to find that spatial memory processing and hippocampal development tend to emerge in infancy at the same time. One of the other things that we know is that LTP and spatial memory tend to be the greatest in younger rodents. And this is because plasticity is more common in younger brains. I've mentioned this quite a bit in class. If you have brain damage, it tends to be a lot better. Not that brain damage of any kind is good, but it tends to be better when you're younger because your brain is much more plastic. In older brains, cells are in the process of shrinking, atrophying, and possibly even dying. And because of that, that means less capacity for learning. Now, we're going to kind of move on to talking about how LTP supports different types of learning that we do. So we're going to focus first on how or whether LTP actually supports associative learning. Now, there are many kinds of learning that do involve associating a stimulus with a response. Like if I see a green traffic light, that means I go. Now, the previous examples that we've looked at don't necessarily involve these kind of associations. So the question becomes, can LTP support associative learning as well? So we're going to start by talking a little bit about classical conditioning. We're going to talk about a very common form of conditioning known as fear conditioning. Sometimes this is also referred to as conditioned suppression. So this is a pretty standard type of classical conditioning. So a rat will be put in a chamber or a Skinner box and will be presented with a tone on its own. So this tone on its own does not produce a fear response. And for this reason, it is what we call a conditioned stimulus. It does not do anything on its own. Now, one of the things that we do know is that if you give a rat a small little electrical shock on its feet, rats will typically freeze. This is a very typical fear response. It is instinctual in nature. So the electrical shock in this case is our unconditioned stimulus. It is a painful stimulus that produces a reflexive instinctual response. In this case, what we would call our unconditioned response. If you're having trouble remembering the difference between a conditioned stimulus and an unconditioned stimulus, think about the fact that conditioned is another way of saying learned. So if I flinch or freeze because I received an electrical shock, that is not something that I learned to do. That is something that is instinctual. So freezing to a foot shock would be our unconditioned response. So what we do for a series of several trials is we briefly present that tone that on its own did nothing, and we pair that with electricity in the floor, the little foot shock. So the rat will freeze as a result. Now, over time, after we present tone, foot shock, tone, foot shock, tone, foot shock, the rat will begin to start freezing in response to the tone on its own. So the tone alone will produce freezing. So in this case, freezing has become a conditioned response. The rat has seemingly learned that when you hear that tone, that a foot shock is about to happen, and so it freezes in anticipation. So this is a really basic example of fear conditioning, and there is some evidence that suggests that long-term potentiation does, in fact, support this form of learning. In particular, we do find that long-term potentiation in the amygdala is very strongly correlated with the freezing responses that a rat will do. And you might be kind of surprised that I mentioned the amygdala when I've been talking a lot about the hippocampus. But I just briefly want to show you that the amygdala is directly anterior to the hippocampus. So the amygdala is connected to the hippocampus. It is an area that we'll talk more about later, but it is very strongly associated with fear. Now, what's interesting is that if I randomly present tones and shocks to a rat with no specific order, so sometimes the shock comes first, sometimes the tone comes first, there's no association that's made. So what's really critical for fear conditioning is that that conditioned stimulus, that tone, actually has to be predicting something for that association to be made. So here's kind of another view. So here is our hippocampus, and here is our amygdala. So the amygdala is located right here. The hippocampus will be just posterior to that. Now, some researchers, so I kind of mentioned this a little bit already, so they've done fear conditioning with rats. So they've tested with a tone. So they conditioned these rats. They tested them with the tone alone. Then during training, they got the tone and shock. And then during tests, they got the tone on their own. Now, they were either trained with the tone always coming before the foot shock, so that's our conditioned group, or they were presented out of sync and in random orders, which is basically a control condition. So when you present them in these random ways, the tone no longer predicts the foot shock. It's random, and so you'll kind of notice that really the EPSPs in the amygdala don't really change all that much. Now, in contrast, take a look at our conditioned rats. So here is our tone alone. So prior to this pairing, notice that long-term potentiation happens both during training and stays after training. So this increased long-term potentiation correlates with the freezing behavior in the conditioned rats. Now, you'll notice that we didn't do any high-frequency stimulation like we did in the hippocampus. But the assumption here is that enhanced activity is being sent to the amygdala, not only from the hippocampus, but from our sensory systems. So you don't necessarily have to deliver high trained stimulation. You can just have activation being sent from the amygdala or to the amygdala from these other brain areas that are also processing this stimulus. So this experiment suggests that co-activation of separate but converging inputs is sufficient and can induce long-term potentiation. So basically, a weak input can sustain a long-term potentiation if it's paired with something stronger. So the tone in this case is our weak input. It does nothing on its own. But if we pair it with something very strong, like a foot shock, that will trigger a larger response in the amygdala, which will be associated with fear. Now, here's what's really critical for this long-term potentiation to occur. The association must occur close in time. So let's talk a little bit about how this might happen. Now, this image looks a little confusing, but I'll do my best to try to walk you through it as much as I can. So let's talk about how this happens. So some regions of the brain are receiving inputs from many different systems: the somatosensory system, the auditory system, the visual system. And as we'll talk about later, the amygdala and the hippocampus are two of those areas. They have a lot of different connections to our sensory systems. Now, some of these connections are very strong. Some of these connections are weak, but can be strengthened. So what you're kind of looking at here are different synapses on a particular cell. Now, in particular, I want you to focus on this particular synapse. This is what we would call a strong synapse. So a strong synapse we've already kind of talked about. This strong synapse, illustrated here, has more AMPA receptors, more NMDA receptors, dendritic spines, which create more surface area, and more glutamate that is released. So what happens in a foot shock experiment? So we can think of the foot shock as being a strong synapse. So if I get shocked by something, that is going to create a very instinctual, reflexive behavior almost immediately. And we could consider that the synapse that's associated with processing the shock is very, very strong. Now, let's take a look at this one, for example. This synapse could be considered a weak synapse. And in this case, that would be the tone. Now, why is the shock input strong to begin with? Well, we're dealing with pain. So we're getting a lot of input from the spinothalamic system. We're getting a lot of pain. And these connections and having a strong synapse for painful stimuli is really important. Animals learn to avoid painful stimuli very, very quickly. So to kind of give you an example, we have a scat mat. Smedley's not allowed to go in the hallway, and it does deliver a tiny little electrical pulse on his feet if he steps on that mat. He has only probably stepped on it twice and now knows to avoid the hallway completely. So animals are really good at learning from pain, and that indicates that that is a very strong synapse. So if I get a foot shock, so if I'm a rat in a fear conditioning experiment and I get a foot shock, that is going to activate that strong synapse. And that EPSP from that strong synapse is going to diffuse to other synapses. So this is what we call kind of a dendritic spike. So in this case, this activation is going to wash down different parts of the other synapses. And that's going to depolarize them a little bit. So you can kind of see that here. So here was our strong synapse, and you can kind of see that that activation is washing down all of the other synapses. And that's going to have the effect of depolarizing them a little bit. So what's going to happen is that input into this weak synapse, so it was weak here, but now that it's been slightly depolarized from that strong synapse, magnesium has been removed from these NMDA receptors. So in this case, those receptors are primed at this point, which means that calcium can now get inside of the cell. And so because this strong synapse depolarized and primed the NMDA receptors with that weak synapse, we get a long-term potentiation in the weak synapse, and it thus, as a result, becomes strengthened. So let's close by talking a little bit about why LTP occurs in the amygdala. Now, that previous picture that I showed you could be used for any region in theory that is necessary for associative learning. The amygdala is just one of these. We do know based on animal research that LTP does occur in the amygdala and it does support fear conditioning. We do know that the amygdala is very rich in NMDA receptors like the hippocampus. NMDA, so if we take that AP5, that NMDA receptor antagonist, one of the things that we find is that if you apply that to the amygdala, that actually prevents fear conditioning from occurring. So if I present a rat with a tone and then a foot shock, the rat will not learn to freeze in response to the tone. And this can be done in animals that are awake or behaving. We do also know that if you genetically enhance aspects of NMDA receptors, this will increase LTP as well as fear conditioning. And these LTPs can be measured in vitro or in vivo.